Imagine if something as small as a bee could take down one of the deadliest diseases on Earth in less than an hour. That is exactly what made headlines when scientists reported that bee venom could destroy certain cancer cells in just 60 minutes. It sounded like the plot of a futuristic medical thriller, but this was real research conducted in a laboratory by serious scientists. At the center of this discovery is melittin, the powerhouse peptide that gives honeybee venom its sting. Let's dive in and unravel how this tiny molecule delivers such a massive punch against cancer cells. In 2020, a team of scientists from the Harry Perkins Institute of Medical Research in Australia revealed a discovery that stunned the cancer research world. They had been studying honeybee venom and its most potent ingredient, a peptide called melittin, to see how it interacted with human cancer cells. Melittin is small but extremely powerful, capable of disrupting the outer membranes of cells by forming tiny pores that cause the cells to lose their contents and die. The researchers tested it against two of the most aggressive types of breast cancer, triple-negative breast cancer and HER2-positive breast cancer. Both of these cancers are known for rapid growth, high recurrence rates, and limited treatment options. In controlled laboratory experiments, purified melittin was introduced to cancer cells growing in petri dishes. Within just 1 hour, the membranes of those cancer cells were destroyed. Yes! Within just 20 minutes, melittin had already begun shutting down key signaling pathways inside the cells, including the PI3K/ACT pathway, which cancer cells use to grow and survive. This rapid, double-layered attack was unlike what is seen with many conventional drugs, which often take days to show significant effects in cell cultures. However, the scientists also acknowledged that melittin is not naturally precise enough to be injected directly into the human body as a treatment. It is a powerful membrane-damaging molecule, and in raw form, it can harm healthy cells, including red blood cells, leading to dangerous side effects. This challenge has led researchers worldwide to explore delivery systems that could target melittin specifically to cancer cells while sparing the rest of the body. One strategy involves encapsulating melittin in nanoparticles or liposomes, which are tiny, protective spheres that release their contents only when they reach the tumor site. In preclinical mouse studies, these targeted delivery systems have been able to slow tumor growth and limit damage to healthy tissue. Another approach is to link melittin to antibodies that recognize specific proteins on cancer cells, ensuring the peptide is delivered only to its intended target. These antibody melittin conjugates have shown promising results in animal models, but they are still far from human clinical use. Outside of research labs, bee venom therapy, also called apitherapy, has been promoted in alternative medicine circles, sometimes with claims of curing cancer. Medical evidence does not support these claims, and there are significant safety concerns. Bee venom can cause severe allergic reactions, including life-threatening anaphylaxis, and there have been documented fatalities linked to its use in uncontrolled settings. The difference between a laboratory study and an unregulated injection is enormous. In the lab, scientists work with purified melittin under precise conditions, measuring doses down to the microgram and carefully observing effects on isolated cells. In a human body, raw bee venom would interact with many tissues and organs, making unpredictable and potentially dangerous reactions far more likely. That is why no cancer treatment based on melittin or bee venom has been approved for use in people, and no completed human clinical trials have proven its safety and effectiveness against cancer. What the research does offer is a foundation for future development. Scientists are experimenting with engineered versions of melittin that remain inactive until they encounter the unique biochemical environment around a tumor. Some designs respond to the slightly acidic conditions near cancer cells, activating only when they arrive at the tumor site. Others are shielded within molecular cages that open in response to enzymes produced mainly by cancer cells. These smart delivery systems could potentially make melittin a precise and safe cancer-fighting tool. There is also growing interest in using melittin as part of combination therapy. By damaging the cancer cell membrane, melittin could allow other drugs to enter the cell more easily, increasing their effectiveness. In laboratory studies, melittin has been shown to make cancer cells more sensitive to certain chemotherapies, suggesting it might one day enhance existing treatments. Another potential advantage is that melittin's mechanism, physically puncturing membranes, could help overcome drug resistance, which is a major challenge in cancer therapy. Cancer cells often develop ways to pump drugs out or repair the damage those drugs cause. But a physical breach in the membrane is harder for them to resist. Even with these exciting possibilities, researchers emphasize that translating laboratory success into real-world treatment is a long, uncertain process. Many compounds that look promising in cell cultures fail during animal studies or human trials because of toxicity, delivery problems, or lack of effectiveness in the complex environment of the human body. Melittin faces these same hurdles. The fact that it can destroy cancer cells in a petri dish in 60 minutes does not mean it can cure cancer in a person in the same time frame. Still, the research is significant because it highlights how nature can offer potent chemical tools that inspire new medical approaches. For now, bee venom's role in cancer research is a reminder of both the promise and the caution needed in translating lab discoveries into treatments. The next steps will require years of additional research, careful testing in animal models, and eventually, well-designed human trials. Until then, it remains an exciting lead, not a proven cure. The story of bee venom and melittin is far from over, and it will be fascinating to see where the science takes it in the coming years. Until scientists complete the work needed to turn this discovery into a safe and effective treatment, melittin will remain an exciting possibility rather than a proven cure. You can like, share, and subscribe for more crazy stories from the world of science, entertainment, and discoveries that could change the way we think about the future.